Hello, welcome to another screencast. This one is on the pre-lab discussion 2 for the bacterial transformation. So at this point, you may or may not have already done the lab by the time you're watching this. But by the end of this, hopefully here you're able to describe and explain what transformation is, talk about the structure and the purpose of plasmids, and so defining what a plasmid is. And then looking at being able to describe and explain here the structure of the plasmid, specifically the one we're using is called pGlo. And so on that plasmid there are genes, so be able to identify what those genes are, and then also be able to describe and explain the expression products of those genes. Then also the purpose of the whole lab activity in general, as well as then each of the 4 plates that you have there. Should be able to describe and explain any predictions you're making, as well as then ultimately here explaining the results. Right, so why do we use the plates? What do the plates show? What's the whole purpose of this activity? So this isn't really so much an experiment as it is an activity to demonstrate some of these principles that we've been talking about in terms of gene expression and then regulation of gene expression. Right? So let's get started here. A little bit of historical background. We talked earlier about the Griffith's experiments here with strep pneumoniae, and then the smooth bacteria here that its phenotype was able to kill the mice, and then these living R ones here that were rough, they were not able to kill mice. So what he did was he took these living S and then heat killed them over here, and then injected that with the living R into the mouse, and the mouse of course died, and then he pulled out here living S. And so what he came to the conclusion was that they were in some way transformed. So he coins this term, and it turns out that something similar to what we are doing was happening there, where you had these are the R, and then the DNA from the S was in some way being incorporated into these R cells, and so now you had S DNA inside there, and then that made them living S, and so then the DNA of the S was being transcribed and translated, and then you're getting the characteristics of then this actually looking like S. So, and it turns out that bacteria are able to do this, and they do this sort of naturally all the time, and we are going to exploit that or take advantage of that or be able to use that in biotechnology. Right? So in general, biotech takes advantage of naturally occurring processes, and then manipulates them, changes them in a little bit to do something that we want with that. Okay, so here we see E. coli, and so this is representative of E. coli, and then this is a plasmid, which I'll talk about in just a little bit. Well, a specific plasmid, but in general, plasmids are these extra genomic DNA, meaning that it's not that this little DNA circle right here is not part of the bacterial chromosome, is not part of the genome. So in this E. coli, this chromosome, there's a single chromosome, and that chromosome is generally circular, and so this whole thing is the genome of the E. coli, of the prokaryote. But it turns out that many bacteria, prokaryotes, have these plasmids, and these are extra genomic. They are small, circular pieces of DNA that have 2 to 5 genes on them. So not a whole heck of a lot of genes compared to the entire genome of the organism over here. Okay. And it turns out that these many of these plasmids encode genes that make them resistant to antibiotics. Right? And so this AMP-R gene right here, this is ampicillin, which is the name of an antibiotic, and then R stands for resistant. So many bacteria are able to have these plasmids, or do have these plasmids, and it turns out that naturally these bacteria are able to take up plasmids, right? And then also get rid of them and exchange them with other prokaryotes as well. So these things naturally normally exist, and we are in biotechnology applications, we're taking advantage of that. So we're going to transform our E. coli in the lab, and here's just a little animation of what we did there. So you chilled this down, added the plasmid to the plus P glow tube, and then you try to make this thing take it in. You heat shock it, right? You put it in the hot water baths. That helps loosen up the wall a little bit. The plasmid then will enter the cell. Then you cool it back down. Little calcium chloride bath, little LB broth, and now that bacterium has been transformed. So let's talk a little bit about the plasmid that we're using here in this lab. So the plasmid we're using is what is referred to as a recombinant plasmid. And recombinant means that it is DNA from two sources. This contains DNA from two or more sources. Right? And so in this case, the plasmid is a prokaryotic plasmid. And we're going to take a gene from a jellyfish, right? And these jellyfish cells are eukaryotic. And here's the gene right here. So in another biotechnology application, which we'll discuss a little bit later, we take enzymes, right, and you're able to cleave or cut out the gene of interest. Right? So we're able to cut this and then isolate that gene right here. And then we use another enzyme to cut the plasmid open, and then this gene gets inserted into the plasmid, and now this plasmid is known as recombinant because it contains DNA from the prokaryote and it contains DNA from the eukaryote here. So in general, recombinant, two or more sources of DNA. Then we're going to use this recombinant plasmid to hopefully put into the E. coli, right, to transform the E. coli. All this stuff up here, all the stuff that I'm describing right here has already been done. We're not doing this in our lab. I bought the kit, I bought the plasmid already transformed. You could also buy a kit that actually does the splicing here, cuts this open and cuts out this gene over here and puts it in, everything like that, but that's time consuming. And so I just cheated a little bit there and bought the plasmid already recombinant. Now, so what are the genes that are on this plasmid? Right? There is a gene for ampicillin resistance, right, which is right here. And so this is a map down here of the plasmid. Right? And so what we're saying is the map means that where is the gene. So this segment right here is the gene. So if you were to zoom in here, you'd see a DNA sequence right here of A's, T's, C's, and G's, etc. That would then, when this is transcribed and translated, will get you an enzyme that will break down ampicillin. Now, ampicillin is an antibiotic that will kill E. coli. So E. coli that are said to be resistant mean that they have this gene that is being actively expressed. When you express that gene, you make this enzyme that will break down the antibiotic, and the bacteria are then able to live in the presence of that antibiotic. Right? Here on this map, they call this BLA. In the lab, we also sometimes refer to it as AMP-R for ampicillin resistance. Right? Also on the plasmid, there is another gene right here. There's another gene right here. This is the ARA-C gene, and this is the regulatory gene. So when this is transcribed and translated, we'll get you a repressor protein. Gets us a repressor protein that is made in an active conformation. So that means that this is part of an operon, or not part of the operon rather, but that this repressor protein will bind to an operator, right? And so this part of the plasmid right here is part of what is known as an arabinose operon. And so we have the promoter here and the operator. Right? And that arabinose operon has been altered to take out the genes that were normally there, and this is where we spliced in this gene right here, which is GFP. So this GFP gene, when this gene is transcribed and translated, will get you a protein called GFP, which stands for green fluorescent protein. Now this protein is special because under UV light it will glow, it will fluoresce green. So if you add UV light to that, it will fluoresce green, which is pretty neat. So this recombinant plasmid is the plasmid that we used, and that is hopefully going to be taken up by the E. coli. Right? So the genes that are on this plasmid, 1 is the AMPR gene, 2 is this regulatory gene here, this ARA-C, and the third gene is this GFP gene that is part of these arabinose operon that was modified. So here's the lac operon again for you to review and compare. So this lac operon is an inducible operon, right? And this is off but can be turned on, right? When the inducer allolactose is present. It makes the active repressor protein inactive, and it turns it on. So here's the arabinose operon, very similar to that lac operon. It's also an inducible operon. We notice here is the ARA-C regulatory gene, transcribed, translated, gets us this repressor protein. The genes of this operon here, ARA-B, this should say ARA-A and ARA-D, but it's not showing up because I had to modify this for some reason. So ARA-A, and this is ARA-D, so not LAC-Y and not LAC-A. So these three genes here, when they're transcribed and translated, get us enzymes that hydrolyze arabinose, that break down arabinose. Right? So this is very similar to the lactose operon. This arabinose is a sugar that was put into the agar on the plates that are labeled LB with ampicillin in them, and also have arabinose. So this is the label, the LB amp ARA plates. Okay. So what we did is actually we kept the in the plasmid. You keep the ARA-C, right? You keep the PBAD promoter, you keep the operator right here, and then we took out the genes ARA-B, ARA-A, and ARA-D there, and replace it with this GFP sequence. Right? So when arabinose is present, here, that's going to turn this active repressor protein inactive, right? And then we'll be able to get RNA polymerase binding to the operator, moving down over the, binding to the promoter, moving down over the operator, and then expressing GFP, right? And so then this is how we turn the arabinose operon on. This is how we turn the expression of GFP on. Turn on. GFP by arabinose. Right? So again, here is the whole situation, right? We're hoping to get this recombinant plasmid into this E. coli, and then there's the plasmid. So the bacteria can be transformed or not, right? So here's what you did in the plus tube. Right? So this was all in the plus Pilo microcentrifuge tube. This is the lid on the microcentrifuge tube. So you put in lots of E. coli, put in lots of these plasmids, right? You put in 10 microliters of the plasmid, and you put in 1 colony, which represents hundreds, if not thousands, of individual E. coli bacteria. So 10 microliters is many copies of that. So when you do the transformation procedure, some of those E. coli will get transformed and some of them won't, just because of luck, right? So the ones that do get transformed, right, and the ones that don't get transformed, so now in the tube, even after all the transformation procedures, so this would be the tube again, right? Visually, here's the lid. So in the tube, you'd have some that were transformed and some that weren't transformed. So you took this from the plus Pilo tube, and what did you do? You put this onto 2 of the plates that you labeled plus Pilo, right? One that had LB amp and one that had LB amp arrow on it, right? And so these are both going to be plus Pilo, right? So if you put these bacteria onto this plate, right, and these ones onto this plate, and then these ones onto this plate, and these guys over here, right, which ones are going to survive? These guys or these guys? Well, the answer is the ones that are resistant to the plasmid, right? Sorry, back up. The ones that are resistant to the ampicillin, right? That's in the agar. How do you know it's in the agar? Because I labeled it there, ampicillin, right? So this ampicillin is what's known as a selection pressure. And what we're doing is we are selecting, using that ampicillin, we are selecting for transformed E. coli. When I say selecting for, I mean if you don't have the plasmid, if you're not a transformed E. coli, I don't want you living on that plate, right? That ampicillin will kill you. So that you are pretty much guaranteed that if you see any growth on this plate, any little colonies growing on this plate, stop that, any little colonies growing on this plate, any little colonies on this plate, on this one, right? All these colonies contain E. coli that were transformed, right? So, finally, wrapping this up, I know this is a long one, but you should be able to take this, and so we're going to have LB amp minus, and then you had LB amp minus, and then you had LB amp plus, and LB amp arabinose plus, right? So what you should do here is be able to then results prediction. So what would you predict? Right? So this is number 1. Number 2, be able to then say talk about the purpose of each plate. Right? So why do you need this plate? Why do you need this plate? Why do you need this one? Why do you need this one? Right? What are they showing you? Right? What does this tell you? And then third, be able to explain results, right? And then verify your predictions. Okay. So when we come back after the weekend, we're going to look at the results here. So you should before then you should be able to predict what you're going to see, and then be able to explain what's going on there. Okay, so you should be able to now describe and explain what a transformation actually is, what we mean by that, the structure and purpose of plasmids in general, be able to describe and explain here the P-glow plasmid, and being able to identify there are 3 genes on that plasmid, and then tell me what the expression products are of each of those genes. I didn't talk about this, but you should be able to now figure out what the whole purpose of this lab is, be able to tell me the purpose of each plate, make some predictions, and then be able to explain the results ultimately when we get back. Okay, thanks for listening. If you have questions, come on into class or Broom Block, and we'll see you later.